Thank you. Hi, everyone. My name is Alex, and I'll be today's host. Welcome to today's Jameel Clinic seminar series, where we'll be listening to Armando Solar-Lezama talk about predictions and explanations in neurosymbolic learning. Take it away.
Thank you. Thanks a lot. So, hi, everyone. It's great to be here. So, I guess I want to start with a little bit of background. So my original expertise actually comes from programming languages. And in particular, in the area of programming languages, my group has done a lot of work around the problem of program synthesis. This is the problem of how do you generate a piece of code that does some interesting thing that you want. And in particular, we have done a lot of work on an approach to synthesis called syntax-guided synthesis, where the idea is you take some constraints that tell you how some piece of code that you want to generate is actually supposed to work. So these are behavioral constraints. You combine them with some constraints on how that code should look like, the structure of the code. Oftentimes, those come from, for example, a domain-specific language that you want to ensure that the programs you're generating come from this domain-specific language. There might be a program skeleton or a program sketch that you want to make sure that the solutions you generate come from that. And so there is now a pretty big community in the within programming languages, looking at how you combine both of these kinds of constraints to generate code, right? And so the field has actually been quite successful. Today, we can do a lot of things that even 10 years ago seemed pretty hard to do. So for example, data structure manipulations, right? The kind of data structure manipulations that you might learn in algorithms class. There's actually not just one, but multiple tools in the community that can actually synthesize even verified implementations of many of these from very high level specifications. One area in particular that has been quite active and quite successful is applications to data wrangling. For example, you have some text data and you want to process it in some way. You want to maybe extract some fields from this text data. If you've ever used the flash fill functionality in Excel, this is actually work that came from the program synthesis community. And this was a project led by Sumit Gulwani at Microsoft Research, actually, already a few years ago. So now you might be wondering, well, what does that have to do with the topic of this seminar? Well, all the way back in 2010, there was actually a paper that was led by Susmit Jha that made this very interesting observation, which is that normally we think of program synthesis as, hey, I have some specification, and from that specification, I want to produce a piece of code, an implementation that implements that. But you can also turn the tables. You can actually take, for example, an implementation that maybe is messy and complicated and difficult to understand. You might not even have access to the code of that implementation. It might be a complete black box. But by observing the inputs and outputs, you can set up a synthesis problem where your goal is to drive that specification, that high-level description of this implementation based on these input-output observations. And so the initial setting where they experimented with this idea was in the context of just obfuscated code. For example, a lot of viruses, a lot of exploits have this obfuscated code that is especially designed to make it difficult to reverse engineer, but you can actually observe how it reacts to certain inputs and certain outputs. And then from that, define a synthesis problem that asks, based on these observations, what is the description what this piece of code does? And from that, very quickly, the community realized that you could actually apply this idea in a number of other settings. For example, there was a paper all the way back in 2012 that showed that, hey, if you want to build models, for example, of how an instruction works in a CPU, this is the sort of thing that you might have to do if you want to create a binary analyzer that is able to analyze assembly code. Well, you could look at execution data from instructions, and then from that, actually synthesize a model of those instructions. We did something similar back in 2016, looking, for example, at the problem of modeling very complex platforms. So, for example, if you think about the Android platform, then this is a giant piece of code. There's even some native code in there. It's very, very hard for traditional program analysis techniques to actually understand what's going on in that platform. And yet, if you want to analyze an Android application, the Android application might be really small. It might be even just a few hundred lines of code. But if you don't know what the Android framework does, it's very hard to make heads and tails of what is going on in the application, because the application relies on that big platform to invoke certain methods in it. And so what we showed was that by running a set of test applications and recording those interactions between those test applications and the main Android framework, we could then automatically synthesize a model of the reactive behavior of the Android application. In other words, a model that describes how control is transferred from the Android platform to the application, back to the platform, that captures some of the design patterns there, but at the same time, that is much simpler than the real thing, and that allows you to actually do analysis in that context. And so what we're seeing in many of these systems is the application of program synthesis techniques to be able to take a system that is somewhat impenetrable, maybe too complex to analyze in detail, but leveraging this ability to generate programs that both satisfy high-level structural constraints and that also match observed behaviors in order to understand these systems. And so it turns out this is actually something very valuable to do, even beyond software engineering. So, for example, within our own group, for example, in collaboration with Wojciech Matusik's group, we actually did some work back in 2018, looking at the problem of reverse engineering CAD models, essentially being able to go from a point cloud describing a solid object, and from that being able to reverse engineer the set of geometric operations that can produce the said object. A different project, for example, led by Kevin Ellis. This was in collaboration with Josh Tenenbaum and Tim O'Donnell, who is a linguist. We actually looked at the problem of reverse engineering language morphophonology, essentially being able to take a corpus of words in a language, and from that derive both morphology rules, the kind of rules that allow you to construct, let's say, as well as phonology rules, the kind of rules that tell you, for example, that the past of walk is pronounced walked and not walked, right? These are the kind of rules that actually make up a language. And it turns out that by framing this as a program synthesis problem, you can actually then discover these kind of rules that in the context of many real languages. More relevant to the topic of the seminar today was actually this work going all the way back to 2013. The student who led this effort was Ali Sinan Koskal. This also included my former advisor, Raz Bodik. And so there in this paper, the idea was, well, if we have this machinery that allows us to reverse engineer a system, what happens if we try to apply it to a living organism? And so in particular, the idea here was to be able to derive some hypothesis about the mechanisms that control cell fate in this case, in the context of C. elegans. And so the idea is that you want to be able to extract a regulatory network where you have simple state machine representations of what happens at every state and what causes that state to trigger and either promote or inhibit the production of a different protein. And so the high-level idea is that you start with some data. In this case, it's data from a large number of experiments that have been conducted over many, many years, where you are looking at, for every experiment, there are some proteins, for example, that are removed by turning off the relevant genes, and you're looking at the cell fate decisions. Essentially, you're looking at the fate of 6 neighboring cells, and in some cases, the exact same setup and the exact same experiment might lead to different outcomes, right? There's a lot of non-determinism in real living organisms. And so the idea was to frame this entirely as a program synthesis problem, even with the non-determinism. The idea behind handling the non-determinism was to model it as being the result of scheduling decisions, essentially race conditions, what happens first. And so what you want is you want a program that represents a model, and that program is a function of both the input, in this case, the particular mutations that you choose at every particular point, essentially the genes that are turned on and off, as well as this non-deterministic scheduling decisions. And so the idea is that the fate, the outcome that you observe, is a function of both the proteins available, as well as this non-deterministic scheduling decisions. And so what you want to ensure is that there isn't a schedule that can produce a fate that was never observed in the experiments. And you also want to make sure that your program has the property that for all the fates that are observed in the experiments, that they can actually be produced by a schedule. And so by framing this as a program synthesis problem, this allows you to actually do some interesting things. For example, you can take the data and you can generate not just a model that is consistent with the data, but you can generate multiple models that are consistent with the data. And now you can actually start asking questions like, can I have, what experiment might allow me to distinguish between one model and the other? How do I know which one of these two models represents reality? Well, I can actually set up an experiment and figure it out. And so in this way, the system can also allow us to characterize the uncertainty behind the data that we have collected so far. So this was, in my opinion, a really cool strategy and a really cool idea. And so the obvious question when you have a really cool idea from a few years ago is, well, why hasn't this taken over the world? And part of the solution is that given the techniques that we have so far, we've as a community, we've been working on this program synthesis problem, or at least on the modern view of program synthesis, for about 15 years or so. And we can do a lot of impressive things, but it's still a pretty limited set of techniques. So for one, you're fairly limited in the size of the code that you're able to produce. You're not really able to produce really big programs. We're really actually extremely constrained using traditional techniques to programs on the order of maybe a dozen lines of code. You can boost that up by applying some domain-specific insights and by playing divide and conquer tricks. But in general, it's difficult to scale these traditional program synthesis approaches. Why is that? Well, it's because you're really searching in this very large combinatorial space of possible programs to find this needle in a haystack, this program that is actually going to match all your data and that is going to match all your constraints on the structure. Often there's a lot of manual effort required in describing the space of possible programs that the technique is going to consider, so that you manage to capture everything that you know about the domain, but at the same time, you still give it enough flexibility to discover interesting things. And so the kind of techniques that came from the programming languages world, they had a lot of these limitations. They also do not work great with noise. They're very good when you want to impose absolute constraints. But as soon as you have to consider noisy data, the scalability really suffers. And so a couple of years ago, we started an effort that is funded by NSF as part of their Expeditions in Computing project to build on some of these ideas and build on some of this prior work, but bring in a lot more modern machine learning and bring in, try to achieve more of a synergy between sorts of things that the fully symbolic methods that came from the programming languages world were good at, and combine that with things for which modern machine learning techniques, and especially deep learning, are good at, in order to be able to attack some of these more ambitious problems. The idea is to really be able to establish a synergy between the logic-based symbolic methods that we have a lot of experience with within our group, and combine that with neural techniques in order to get, in some ways, the best of both worlds. And so over the past couple of years, we have explored a few different avenues and a few different strategies for combining neural and symbolic reasoning to try to leverage the best of both worlds. For example, in some cases, you want to leverage the ability of neural networks to associate meaning with very ambiguous, potentially unstructured inputs, to translate that into more structured, more symbolic problems that then you can solve using symbolic techniques. In some cases, you actually want to leverage the ability of the neural network to learn very complex conditional distributions over things like programs, in order to be able to directly map from a goal to a program in a much more directed way, in order to be able to get bigger, more interesting programs. In some cases, what you really want is to first leverage the ability of deep learning techniques to learn complicated functions with potentially complicated fitness criteria. And then once you've learned that function, extract from that an interpretable model that then allows you to explain the mechanism behind the observations that you made. And finally, in some context, it's actually very beneficial to really generate models that have some symbolic aspects, and that in those symbolic aspects, they're able to capture domain knowledge and background knowledge about the domain, but that also internally leverage deep learning techniques in order to make sure that they actually get the details right, and in order to give you the ability to actually learn complicated mechanisms. And so in the rest of the talk, I want to focus on a particular project that has emerged as part of this collaboration. We've been really very fortunate to be able to work with our colleagues in the biology department. Philip is actually here in the room, Chris Burge, Kyla McHugh, who's a graduate student there, as well as some of our colleagues from the program synthesis and neurosymbolic learning communities. And so this is still an effort that is, I would say, at an early stage, but I would like to share some of what we found in exploring this problem of RNA splicing using neurosymbolic techniques. So what is it that we're trying to do here? So we are trying to understand how RNA is spliced. So when RNA is produced from a gene in DNA, when a strand of messenger RNA is produced, there are some fragments of that RNA that actually correspond to what is actually going to be expressed in producing a protein, and these are known as exons. And there are some other fragments that have to be spliced away. These are going to be the introns. And so our goal is to understand exactly how this splicing mechanism happens and what determines where a particular gene is going to be spliced in different situations. And so a very high-level framing of this problem is that given an input sequence of RNA, and given if this RNA is going to be spliced in this particular way, then what we want as output is a signal that tells us where the beginning and end of different introns or exons are going to be in that sequence. So essentially what we're looking for is a mapping from positions in that sequence to a value that tells us whether this RNA sequence is going to be spliced at this particular point. And so this is a fairly well-studied problem. And a few years ago, there was actually a paper that showed that if you throw deep learning at this problem, you can actually do a pretty good job at predicting some of these splice points. So you can actually train a neural network to take a large window from a gene, and then from that make a prediction about where the splice points are going to be. And the top K accuracy that you can get here, so in this context, top K accuracy refers to the accuracy that you get when you calibrate this to produce a number of splice points that corresponds to the known number of splice points in a gene. And so from this, it might seem that, well, the problem is solved. We can go home. We can train a neural network to make these predictions. It can make them pretty well. So what's wrong with that? Well, the problem is really a fundamental problem whenever you're trying to follow this end-to-end learning approach in the context of scientific discovery, which is that maybe the network has figured out how splicing works, but we haven't figured out how splicing works. And what we have here is a black box model that doesn't account for anything we know about the splicing mechanism. And for that reason, it becomes difficult, for example, to do different kind of interventions or to evaluate how different kinds of interventions are going to affect the splicing process. Sure, we can evaluate how, for example, mutations in the sequence might affect splicing, but we can't really do much beyond that, right? The model doesn't really improve our knowledge of the underlying system. And so in order to understand how we might be able to do better, it's useful to give a little bit of an overview of what is that mechanism that we would like to understand, right? And so at a very, very high level, essentially what you have is if this is your exon that in your gene, what you have are a set of motifs, this very characteristic sequences that identify the beginning and the end of that exon. And so these are fairly well-known motifs, fairly well characterized. And so in principle, you would think that, well, we just go look for these motifs, and that tells us where the exons are. The problem is that these motifs are too short to have really enough information to actually characterize the where splicing takes place. And so what you find is that, yes, you see this motif at the beginning, at the end of the exon, but you also see it in lots of other places that are not actually the beginning and the end of these exons. And so now the big question is, well, how does the cell know which one of those are the real splice points and which ones are not? And part of how it knows is because there are these regulatory elements that attach to very specific sequences and that have the effect of either inhibiting some of these splice points or enhancing some of those splice points. And so the combination of the effect of all of these different splicing regulatory elements is what actually helps determine where this gene is going to splice. And so we know a lot about these regulatory elements. We know a lot about the proteins involved. We know a lot about the kind of sequences they attach to. None of that is captured if you're just training an end-to-end model from sequence to the prediction. And so what we want is we want a model that actually describes splicing in terms of these regulatory elements. And so how might we go about doing that? Well, here's a simple idea. What we can do is take these motifs for these individual regulatory elements, and these have been characterized by in vitro experiments, looking, for example, at what sequences different proteins attach within RNA. And so we can actually take these known motifs and scan the sequence to identify at every position which motifs might be present, right? And so then we can train the model. Instead of training it directly on the input sequence, we can train it on the results of running all of these motifs through the sequence. Turns out if you do that, though, you get a accuracy that is much, much lower than what the end-to-end model is able to get, right? So it means that we are missing something when we do this, right? These motifs by themselves are not really capturing enough information in order to do a good job of making these predictions. And so now we can start making some hypotheses, right? So maybe we're missing some important regulatory elements, right? Maybe there are some additional motifs that we don't know about and that are actually really important for the process. Or maybe there is some important information that cannot really be captured by these local motifs. For example, secondary structure in how these RNA faults that might be very important for splicing. And in fact, we know that this can be important for splicing, or maybe actually these motifs that have been created, maybe they're not quite right. Maybe they're close, but not quite, not sufficiently precise in order to allow us to do a good job of prediction. And so now we can actually start trying to rule out some of these hypotheses. For example, we can build a model where we have all our known motifs, but we also allow it to discover some brand new motifs from scratch in order to allow it to do a better job making predictions. Now, we can do this and see what kind of accuracy we get, but this starts creating some problems, right? And this is where the learning problem starts getting interesting, because it's very easy for the learning mechanism to essentially leverage these additional sort of free motifs and to just use them to encode the sequence and just communicate the whole sequence to the downstream model, the model that is trying to make these predictions. And so you might get something that, you know, it might look like a motif, but in reality, all it's doing is communicating the entire sequence to the downstream model. And so then you get something, sure, it might give you very high accuracy, but doesn't actually correspond to anything physical. It's just passing information to the network and allowing the network to figure everything out from scratch, potentially even completely ignoring all the motifs that you already have. And this becomes a big problem whenever you're trying to do this essentially interpretability through modularity, right? You want to decompose the problem into these different modules that actually have a clear meaning with respect to the mechanism that you're trying to model. But now, how do you actually enforce that these different components in the model actually correspond to the things that you want them to correspond to in terms of the physical world? And in this case, what we found is that if you impose an information bottleneck in these in terms of how much information, how many actual bits of information these new motifs are able to contribute to the downstream network, what you find is that you can allow them to transfer lots of information, and then you get a really high score, but now they're just communicating the whole sequence. As soon as you start imposing that bottleneck and restricting how much information they can transfer, all of a sudden we find that we cannot train them to get higher accuracy from what the original model was giving us. And so what this suggests to us is that just adding a few additional motifs is not actually what is the problem in this model. And so now we can move into a different hypothesis, right? Maybe these motifs are close, but not quite right, and not quite accurate enough to allow us to predict splicing, right? And so how do these filters actually work? So essentially, we have these motif models that were computed from RNA bind and seq data. And so these RBNS motifs, they're fixed. They come from a different set of experiments. And essentially, we just take the output of this model, we pass it through a threshold to filter the signal and impose an information bottleneck. So now what we're going to do is we're going to generate a model that is a little bit more complicated. What does this model do? Essentially what it says is it looks at the original motif model, but it makes this filter essentially twice as permissive so that it can capture more potential binding sites, right? So even places that the original model deemed to be plausible but very low strength, it still says, well, let's identify all of those. And then we train a neural network to compute a score only for those locations, right? Only those locations that the original motif model said, yes, there is there is something here, there is some signal here, but now we let the neural network actually figure out what that signal is going to be. And we do that before passing it through the final filter. And so what that means is that there might be some locations that the original motif thought they were pretty strong, and the new model with adjusted motifs decides that no, actually, it's not strong enough to rise above the threshold. And there might be other locations that the original motif said, no, this is too weak. It doesn't rise above the threshold. But the neural network can decide to actually push its score higher and make it actually be above the threshold. However, what the neural network is not allowed to do is take a location that the original motif model regarded as being just completely irrelevant and make it relevant. It can only restrict itself to those locations that the original motif models already deemed important. And so what we have now is we have this model that is going to be trained end-to-end to discover both these adjustments to the motifs as well as the downstream model. And what we find is that when we do this, we can actually get significantly higher accuracy compared to the model that was using the original motif. But now, of course, there's a big question. Are these new adjusted motifs real? Actually capturing something physical, something in the actual mechanism? Or is our network just leveraging this new neural component in order to cheat and to transfer some additional information in a way that doesn't actually match the underlying biology? So that is one challenge and one thing that we need to figure out. The other challenge is that as soon as we have this combination of, essentially now we have branches, we have these different kinds of information bottlenecks where we're actually limiting the information in terms of a number of bits, for example, that are able to transfer. All of a sudden, the learning problem becomes significantly harder. All of a sudden, we now have a learning problem that is full of local minimas and that becomes much harder to optimize around. And so this actually required the development of different algorithms in order to be able to do this training effectively. I'm not going to go into details on that. For this talk, I want to talk more about this question of how do we get to the bottom of this question of whether these new adjusted motifs are actually, quote unquote, real. They actually correspond to something real. And so I'm going to describe a couple of experiments that we have done so far that, give us some evidence for this approach actually capturing something real about the process. So at a very high level, essentially what we have, so in the original Strawman solution, essentially what you have is from the RNA bind and seq data, you get this initial set of motifs, and then you train a model that first encodes the sequence using only these motifs, and then is looking to train these downstream models. So we call this the FM downstream model because it's trained from these fixed motifs. And then alternatively, we have this new improved model where in addition, where you also start with the original RBNS motifs, but you are adjusting them. You're learning these adjustments to the motif, and you're also training this downstream model. And so the question says, well, could it be that this combination of the adjusted motifs and downstream model are just figuring out some sneaky way to transfer information about the sequence? And so one of the experiments we did was, well, first, essentially, if you compare them side by side, you have something like this. So first, we binarize the output of these motif layers. Essentially one bit of information for every motif telling you whether that motif is present at that location or not. So this significantly restricts the amount of information that can be transferred from the motif layer to the downstream model. But more importantly, it now makes the two models compatible, right? Now you have a clear interface of exactly what these bits means, and you have the possibility of actually exchanging them. And what we find is that if you take the original downstream model that's trained from the original fixed motifs, and you now run it with the adjusted motifs, it actually improves its performance. It makes sense that it wouldn't be as high as the one with the downstream model that actually was trained with the better motifs. But at least what this suggests is that these adjustments are not completely changing what each one of these signals means relative to what was there before, right? So the fact that we can mix and match between these two models is giving us some evidence, at least, that the strategy for training these models is actually still preserving the meaning of each one of these signals. The other question to look at is, well, so now we have these adjusted motifs. How do they actually compare with the original RBNS motifs when you look at them in the context of the data that was used to train and to generate the original RBNS motif? So this is a completely independent data set, different from the one, the end-to-end data that was used there. This is a data set that essentially contains describes synthetic sequences of RNA and how those sequences and how proteins bind to those sequences. And essentially what we find is that these adjusted motifs actually show a very, very small, I wouldn't make a big deal about this, for all practical purposes, they're behaving the same as the RBNS motifs. But what's really significant here is that, if we look at what's going on here, essentially we had a set of motifs that were generated from these data. And then we have now a new set of motifs that do much better in the end-to-end task, and yet do no worse on this original task. In other words, what that is telling us is that given the original bind and seek data, there's actually different motifs that fit that data equally well, but that actually do quite different when actually run in terms of the end-to-end model. And so I think this also in some ways validates the approach that is still maintaining these new motifs anchored to the original motifs that were there before, but allowing them to drift just enough to actually better capture what is necessary to make end-to-end predictions. Another experiment is what happens if you actually look at a completely different data set, right? So in this case, this is a different data set that also contains information about potential binding sites for proteins that was collected in a very different way. And I'm not quite the expert to be able to describe exactly how they were collected. But again, going back to comparing how the two motifs do in this different data set, we find that the adjusted motifs that were adjusted based on this end-to-end goal In this case, they're actually doing much better, or at least meaningfully better than the original motifs that were computed directly from the RBNS data. So this again provides some evidence that by this technique that we are actually capturing something that is consistent with the underlying mechanism, that it's not just the neural network figuring out sneaky ways of transferring information to the downstream network. And so this is more or less where we're at this point. I think a couple of takeaways that I think we can already talk about. So the first one is, it's very important for learned models to capture prior knowledge about the process we are modeling. Sometimes it is presented as a dichotomy of, well, we can let the data speak for itself and let the neural network figure out whatever it wants, or we can go and painstakingly build models by hand. And you can do both. You can actually leverage a lot of the domain knowledge that you already have and a lot of the data from many of different sources, and but still have the ability to get as much as possible from the data itself. I think another really important idea is this idea of interpretability through modularity. Right? This idea that if you're able to break up a model into meaningful components, and very crucially, if you're able to actually enforce certain semantics around those components, enforce boundaries around those components that make them, the functions that run for the components actually correspond to meaningful parts of that process, that all of a sudden gives you a lot of interpretability. It allows you, for example, to make interventions, not just end-to-end, but also make interventions about intermediate steps in the process. It allows you to probe, for example. It gives you a lot of power that you don't get with fully monolithic models. But it's not easy. Moving from end-to-end modeling does require more sophisticated training strategies. So the moment you start imposing additional constraints on these functions and forcing in certain interfaces and certain structures on them, the optimization problems do become harder, and you have to do some clever things. So to conclude, I think there is a lot of benefit in the synergy between ideas and insights coming from the programming languages and programming systems community and ideas in machine learning and AI that can be really beneficial, especially in those situations where you care about interpretability, where you care about being faithful to your prior knowledge, and where your ultimate goal is not just making predictions, but gaining understanding. And so with that, I'm happy to take any questions.
I think we're going to take questions from the room first, and then we'll take questions from the online participants. So if anyone has a question in the room, all right.
Very interesting. We've been talking about this weekly for, well over a year. But to see it organized in a high level makes you think in different ways, and you've picked certain parts of it to emphasize. But the parts you've picked suggest to me that and this would be intuitively consistent with what we know about these processes and these RNA binding proteins, that the RNA binding protein sequences are really part of the specificity for the process. But when you allow the adjustment model, you're scanning sequences beyond the single RNA binding specificity, And in those flanking sequences, there's information that influences the binding of that RNA protein at that site. And therefore, when you compare just the sequences to each other, you get no insight. But when you compare it to the cross-linking or the data where the latter you did, which is cross-linking of a protein to a specific site on the RNA, you actually see improvement because it's taking into account that signal integrates local information for putting a protein there. So I think we ought to think about through that process and see if we can find a way to identify that adjacency that's adding information.
Yeah, I think we're sort of really only in the first step, I think, of what I think is a lot of potential for actual understanding based on, why are these models doing better, right? And exactly what is the information that they're taking into account that maybe the original set of models was not. And I think in those differences and in looking at the impact that those differences make, I think we can learn a lot.
So you mentioned in the adjusted models, adjusted motifs model, that there is an information bottleneck. I'm curious, like, how is this information bottleneck described, or what do you mean by that? So
actually, Kavi is probably the best person to answer that question. We have the expert in the room. Why don't you come here?
So traditional information bottlenecks in machine learning are usually based on noise. So what you do is that you take a layer and then you'll inject noise. So you'll make sure that it has certain signal properties. So the signal can only be so big in magnitude, and then you'll inject noise of a certain magnitude. This promotes redundancy. So that's the primary goal, is that you promote redundancy. So even if you might have, say, 512 channels, you're only actually able to use, 20 channels worth of information. That's the opposite of what we want here. So in our case, we actually want to reduce redundancy as much as possible, because redundancy makes explaining things harder. If the same thing can be represented in 5 different ways, then you need to understand all 5 ways it can be represented. So instead of that approach, where we use noise primarily, we're instead using the sparsity itself as a way to enforce an entropy bottleneck. So if you think about it, let's say that you've got 1,000 possible positions. If every position can be a 0 or a 1, then that's 1,000 bits of information. But if you only allow, say, 5 of those positions to be a 1 and everything else needs to be 0, then you've got 1,000 choose 5, the log of that. It's going to be a much smaller number of bits of information, something closer to 30 bits of information or something. I don't know the math off the top of my head, but that's effectively how we're doing it. So by enforcing a sparsity constraint on our the output of our motifs layer, we're able to enforce effectively an entropy constraint. Like a regularization there on the wall? So how do we do it specifically? So we tried doing regularization term on the loss. We couldn't get that to work. So instead what we do is that we do a batch normalization followed by a thresholding. And that also doesn't work if you train it directly, or it does, but not very well. So what we do is we start out with a very low threshold, a very high threshold. So we allow almost everything through. Low threshold. Anyway, we allow almost everything through at the beginning. And then as time goes on, we test out our model on a validation set. And if it gets higher than a certain amount of accuracy, we drop the amount of information we're letting through. And we continue to do this, and you get a curve of like the accuracy versus the sparsity versus time, or sparsity versus accuracy. And eventually you've got this like very highly sparse model, and so it's able to work with a very low amount of information being let through.
Thanks. So I know that biology is just one of the domains you're looking at in this expeditions, and I was wondering, what you think the sort of takeaways from this project are that could be applied elsewhere as well. Like, for example, the conclusions you mentioned certainly seem like they're general lessons that would apply in other domains as well. But I was curious if there were any like techniques or like technical contributions from this work that you think could be applied elsewhere as well.
Sure. I think in particular this idea of interpretability through modularity, and for how to enforce modularity and enforce these boundaries in a way that actually forces different components to actually have meaningful semantics relative to what you're trying to process. I think that's a very powerful idea that I think potentially applies in many other contexts. Do you have any questions online?
Does anyone have any questions online? Maybe not. All right. No, I don't think anyone else has any other questions.
All right. Well, in that case, thanks everyone. And